Okay, here we go again. We're going to finish off some molecular terms that are used in cancer. You want to make sure that you can write these down, explain them in about 2 sentences, and then explain the relevance to either the cell cycle or cancer. So let's do kinase, for example. I would say kinases are protein enzymes that add phosphate groups to different molecules. They're important in cancer because phosphate groups can inactivate or activate cell cycle drivers, which pushes the cell cycle forward. Dysregulation of those causes cell cycle dysregulation, which is the basis of cancer. Okay, so we ended on nuclear receptors. A quick note on signals. There's a difference between growth signals and mitogens. So mitogens are signals that cause a cell to divide, whereas growth signals are signals that tell a cell to grow in size. It's just important to differentiate the two. Most of the time when we're talking about these, we're talking about growth signal. We're talking about mitogens, so we're talking about the cell division rather than getting cells bigger. This is Scatman John. He's just an amazing person, and you should listen to his video, The Scatman. Okay, so we talked about transcription factors and nuclear receptors in the previous video, and so right here what we're going to talk about is hormones. Hormones are really simple. They are chemicals that travel from one part of the body to another part of the body, usually involving the bloodstream. Hormones are based on their function and not on what they're made. So hormones aren't a class of proteins or a class of lipids. They are just molecules that travel from one part of the body to signal another part of the body. And some examples are leptin, which signals the amount of fat in your body to your brain; ghrelin, which signals the amount of food in your stomach, or actually the size of your stomach, to your brain; insulin, which signals the amount of sugar in your blood to your brain. And there are different types of hormones. There are autocrine hormones, there are paracrine hormones, and endocrine hormones. So autocrine hormones are going to be released by an area, and they're actually going to come back to that same area. So they can be kind of like, you can think of that as like self-signaling. Paracrine hormones are going to be signals that act basically in the vicinity of the cell producing the endocrine cell, so the cell that is producing the hormones. And then endocrine signals are ones that are going to travel from one part of the body, for example, the liver to the brain, and that's what we are normally talking about when we're talking about hormones. Important note, cells are considered deaf to most hormones unless they have receptors for them. Transcription factors you should be pretty familiar with right by now. A transcription factor is going to be any protein that is going to influence either positively or negatively transcription. So they are going to recruit RNA polymerases or help form this scorpion tail that we've seen in transcription. Please note, so again, the promoter, the regulatory elements, the part of the DNA that tells us when and how much of a protein to make, is what the transcription factors generally bind to. That region is huge, generally involving thousands and thousands of bases before the actual start of transcription. You have activator proteins, which bind to regions on DNA called enhancers, and then you can have insulators that prevent that. And promoters, which are going to help RNA polymerase bind. Yeah, I'm not going to do any more on that. This is a picture, this is a 3D rendering of a transcription factor. Here you can see the bottom of the transcription factor is sequence specifically binding to the DNA in the major groove of the DNA. I think it's kind of cool. All right, so mitochondria is the powerhouse of the organism. So I think one way of thinking about it is like this. There was a small organism, and it used oxygen to basically burn glucose and was able to generate about 27 little pieces of glucose or ATP from that glucose. While the rest of the world is using a process called glycolysis, which is a chemical enzymatic breakdown of glucose. And basically what we're doing is breaking it in half, making it a complicated molecule, the glucose molecule, into two smaller molecules, and therefore releasing about 2 ATPs worth of energy. So cancer cells are actually going to switch between aerobic respiration and anaerobic respiration. Sometimes it's actually going to be advantageous for them to burn off a ton of energy. So they don't want to be, so it will be in their best interest if they are inefficient. And that's when they're going to be using anaerobic respiration. Again, no oxygen, low ATP yield. High oxygen, high ATP yield is going to involve the mitochondria and the process of aerobic respiration. Okay. If you see a diagram like this, this is not what we are studying. This is a really common misconception in biology, and that is that the process of cell division is also called mitosis. So if you hear, for example, the major parts of the cell cycle are prophase, metaphase, anaphase, telophase, that's not accurate. What most people refer to as cell cycle is actually nuclear division. So here you'll see this cell is actually dividing its nucleus. It takes the contents, the DNA, organizes it in the center of the cell, splits the DNA up into 2 parts, and then puts them into 2 new nuclei, after which the cell divides. Now this is a very, very small part of the cell cycle. So if I were to take all of this entire thing, and put it in the terms of the cell cycle, what you would see is that it would fill this yellow piece here. The actual cell cycle is much bigger and involves far more than prophase, metaphase, anaphase, and telophase. So let's start. Generally, cells are going to start the cell cycle and they're going to receive a signal. That's going to be generally a differentiation signal or a divide mitogen signal. So if it's a differentiation signal, the cell will go into senescence, or G0, which is a state in which cells aren't dividing. And that's actually the state in which most of your cells are. G1, the first stage of cell cycle, is going to be what cells are going to be driven to when we have a mitogen. And what is mainly being done there is you're really building a lot of the machinery necessary for DNA replication in G1. So you're going to have activation of transcription factors that are going to cause the transcription of molecules like helicase, DNA polymerases, topoisomerases, ligases, all of those. And then those proteins are going to assemble on DNA kind of in these replication complexes, and they're going to stay like ready to fire. So basically once S hits, those replication centers fire and DNA starts getting copied. At S right here, DNA is copied and there's going to be some checks. We want to make sure that the sides of DNA are kind of parallel to each other and of equal distance. If they're not, then that indicates we have some cell damage or DNA damage. And one other thing is we're going to make sure that we've copied all of the DNA, but only copied it once. Copying too little of the DNA or double copying the DNA are both pretty bad for the cell. They generally don't end up with good outcomes. So once DNA is copied and it is checked, the cell moves into G2, which is basically prep for mitosis. That's the thing that we were talking about earlier. In mitosis is going to be driven by the M-cyclin CDK complex, which you saw in one of the first slides, and it is going to cause the M-cyclin CDK complex is going to cause the nuclear envelope to disappear. It is going to cause microtubules to arrange DNA in the center of the cell. And then it is going to cause, well, that's, I guess, mainly what it causes. Manu Chao is a guy who makes a lot of songs. He raps in Italian, Spanish, and English, as well as French, actually, and is an important aspect of studying cancer. He will be playing in the background quite a bit when we are listening to, or when we are working on cancer. All right, so you are all familiar with microtubules. You guys have seen this video before, and this down here, this track that this motor is walking on, is a microtubule, and they play a hugely important role in cell cycle in cancer. So In cell cycle in cancer, one of the things that happens is there are these like little points of nucleation, these little circles, and they shoot out microtubules, and those microtubules attach to DNA and align the DNA up in the center of the cell. And when they line the DNA up in the center of the cell, it can be divided evenly amongst the two nuclei. And so that process is going to be heavily regulated by microtubules. So this is kind of what they look like. This is this is one of those microtubule organization centers. Actually, maybe not. It resembles one. And these yellow pieces are going to be the actual microtubules. Yeah, I think so. And these are going to be responsible for many things. Like I said, the lining of DNA in the middle of the cell, but also a big part of the cell structure is going to be determined by microtubules. So this is what it looks like when microtubules are forming. You can see the tips of the microtubules here, and they will actually go and then sometimes fall apart. And they actually do that on their own. This is the microtubule, the role of the microtubule in DNA. So I know this is a crazy image and it's a real image, but what you're seeing is microtubules grabbing DNA and then pulling it apart. Okay, so here we go. I'm going to try to time it so that it does it. Grab, pull apart. Grab, pull apart. Grab, pull apart. Grab, pull apart. This is a pretty amazing video. A lot of people have been waiting a long time for this. So microtubules are also involved in one of the most dangerous aspects of cancer, and that is metastasis. Metastasis or metastasis is a function of cancer cells in which they can leave the tissue they're in and invade other tissues. So in order to do that, basically they have to shoot a part of their cells forward and then destroy the backside of the cell. So they shoot some stuff forward and then suck the back up. Shoot forward, suck it back up. And you can kind of see that happening in the cell here. If you look right at the tip there, you see those new things being formed. They're called filopodia. And then you can see them kind of being taken up in the back. And this is a real picture of a cancer cell that is moving towards a signal. We're going to be talking about lamins and the nuclear envelope a little bit. I'll show you a picture of them here in a second. But lamins are basically going to be a net-like structure that exists under the nuclear envelope and allow it to give it structure. And without the lamins, the nuclear envelope will fall apart. The microtubule organization centers and the lamins are both phosphorylated by the M-cyclin CDK. So the M-cyclin CDK is responsible for, first of all, the lamins falling apart and actually the microtubules increasing in activity and grabbing the DNA and organizing it into the center of the cell. This is what the lamins look like. You can see them stained in white right here. You can see them dissolve, form, be there, dissolve, and reform. Okay, so let's try to time this. Okay, here we go. Formed, formed, dissolved, formed. Formed, dissolved, formed. Formed, dissolved, formed. Formed, dissolved, formed. Anyway, that is basically it. In order to organize the DNA, we have to get it out of the nuclear envelope. And also a cool kind of thing, this is one other function of the M-cyclin CDK complex. It actually activates these proteins called cohesins and condensins, which cause DNA to not crumble up, but scrunch up really, really tightly. So tightly, in fact, that it cannot you can't do any expression. So I just kind of think it's a neat fact, but M-cyclin CDK complex, when active, basically shuts down DNA. So let's go over these really, really quickly. Kinase is going to be an enzyme that adds a phosphate to things. Phosphatase is going to be an enzyme that takes phosphates off of things. Receptors are cell surface proteins that are going to allow a cell to listen to its environment. A secondary messenger is often what a receptor is hooked up to, and this allows the receptor to hook into another pathway, for example, a cell division pathway. GPCR is an example of this. You'll see a lot of GPCRs involved in cancers. Cyclins and CDK complexes come together to form these kinases that are specifically used in the cell cycle. They fall into classes like the G1S cyclin, the S cyclin, the G2 cyclin, and the M cyclin CDK complexes. And when they are named, you just might see them named as B, C, D, and you can look up a chart and see which type of cyclin those are. Hormones are going to be signals that start in one part of the cell, a body, and end up in another. Nuclear receptors are going to be compounds that are inside the cell that bind to a molecule that has traversed the cell membrane and then goes and binds to DNA. So estrogen is an example of that. The estrogen receptor with estrogen binds inside the cell after estrogen has traversed the cell membrane and then goes to DNA and affects gene expression. Transcription factors are going to be along the same lines, but they're going to be proteins which, when phosphorylated often or activated, go directly without a combined secondary part and affect gene expression. Mitochondria, the energy domain of the cell, is going to be responsible for aerobic respiration, which is a highly efficient form of respiration using oxygen. You can think of it as burning. Glycolysis, on the other hand, is going to be anaerobic respiration, which is going to be breaking glucose in half using few enzymes, and generates generally about 2 ATP, whereas aerobic respiration with oxygen, the burning, generates about 27. The cell cycle is the process by which cells divide. It actually has 2 offshoots. One is called G0, and the other you know of as cell suicide. So that's the 2 ways, basically, cells can get themselves out of the cell cycle, and most cells are in G0, not in the cell cycle. G is a stage of the cell cycle. It stands for gap. It's gap 1. In gap 1, basically, synthesis machinery is being created. Synthesis is replication and requires replication machinery, obviously. G2 is set up for mitosis, the M stage, and basically what you're doing is setting up a lot of the M cyclin CDK components. Cytokinesis is the actual splitting of the cell. You guys have seen this kind of mechanism before. Basically what you'll have is a cell. It'll, on the inside, will kind of have this lasso mechanism, and then that lasso will shrink and the cell, 2 cells will pop off, kind of like bubbles. Lamins are the things that support the nuclear envelope. They need to be removed in cell cycle so that the DNA can be split and organized. Mitochs and microtubules are going to be the anchors and tubes which shoot off to organize the DNA, form the main structure of the cell, and also provide the highways for the walking molecules. And then we have nuclear envelope, which is going to be supported by lamins, which is a highly selective membrane that protects DNA from the environment. That's all.